everyone, and welcome to 4-H Online today. Today we're going to talk about embryology. So we're going to look at the development of the embryo over time while it's developing inside the egg. And then we'll actually dissect open an egg and look at the different parts and talk about the functions of the egg. So stay tuned. We'll be right back. Welcome back. My name is Heather Conroe. I am a livestock specialist with the University of Missouri Extension. So my job is to provide educational programming for producers pertaining to livestock. I also have a huge passion for youth education, and so I was very excited to be asked to provide this program for you today. Be sure to check in at 5 o'clock and watch the daily live hatching that occurs daily at 5 o'clock.
First off, let's talk about the difference between the eggs that we hatch and the eggs that we eat. The eggs that we eat will not produce a chicken. It doesn't matter how long you incubate them or watch them, no chick is going to hatch out of those eggs. The average consumption per person in the United States per year is 245 eggs. That is a lot of eggs. How many of you guys think that you eat that many eggs in a year? The eggs that hatch, before they are laid, the egg is fertilized. The zygote, which is what we call the baby in its early divisions, divides and begins to grow. So the 2 cells become 4 cells. Those 4 cells each divide again, giving 8 cells. Those 8 cells divide again, giving 16, and so on and so forth. The embryo will nearly stop dividing until it reaches the correct temperature and humidity in an incubator. This diagram having a little bit of technical difficulties. This diagram shows the parts of the egg, and this is what we're going to do in our activity a little later on is dissect an egg and look at those different parts and talk about their functions. So the yolk is the yellow center found here, and this is what we see that turns yellow when we scramble our eggs. The yolk is high in fat, carbohydrates, and vitamins, and the hen produces the yolk during the day. The germinal disc, found right here, is the point of fertilization, and this is where the zygote is actually going to, or the embryo is going to actually develop. The vitellin membrane is the membrane that holds the yolk together. And so when we say that, hey, I broke the yolk, or mom says, sorry, honey, I broke the yolk, it's not actually the yolk that she broke. It's actually the vitellin membrane. So next time that happens, you can correct your mom. And then next we have the Kalasa. The Kalasa here is a fibrous material. It's kind of like a rope structure on both ends of the yolk. And their job is to suspend that yolk inside the shell so that it's not touching the sides of the shell. The shell is made up of mostly calcium carbonate and has thousands and thousands of tiny little pores in it for gas exchange. So carbon dioxide, what we breathe out, goes out of the egg, and oxygen and moisture move into the egg. Then going up here to the air cell forms at the blunt end, or not as sharp as the pointed end, but the blunt end of the egg. And this pocket of air, it will actually be used by the embryo just before hatching as a source of air. And it does increase in size as the egg ages. The shell membranes inside the egg, there's 2 of them. There's an inner and an outer shell membrane. These surround or contain the albumins. So the albumin is the white portion. When we fry it, it turns white. That's the clear fluid around the yolk when it's not cooked, and then it turns white. The albumin provides a protective barrier against bacterial penetration, and it's the 2 layers that the air cell forms in between. So the thin albumin is nearest to the shell, and it spreads out quite thin, and we'll see that when we dissect our egg as well. Then we have the thick albumin, which is closest to the yolk, and it is it's high in riboflavin, and it's almost pure protein. And we'll see the difference when we dissect the egg. But this is the thick albumin, kind of stays all together, kind of rises up above the thin albumin. The thicker the albumin, the thicker the thick albumin is, the better quality of egg it is. And again, we're going to see this again as we go through our activity. So now we're going to go through the journey of the embryo as it develops day by day. So we're going to talk about how this embryo develops, what's going on every day as it grows. So on day 1, the journey begins. At 21 hours, the brain and nervous system begin to form. And right here we see that germinal disc. So it is starting to form. And at 24 hours, the eyes are already starting to form. Day 2, it's all about the heart. And again, so we see that germinal disc getting bigger right there. So at 25 hours, the heart begins to form. And then at 35 hours, the ears are actually forming. And at 42 hours after fertilization, we have a heartbeat. So this video is actually going to show the heartbeat inside the egg. Now this embryo is still developing inside the egg, and we're going to see the heartbeat at about day 6, where it is bigger and much more clear to see. So let's watch this video as that chick, there's that heartbeat just thumping away. Again, that's about a day 6 embryo. Here we are about day 8. We see the little eyeball, little wing bud as he's moving around. Here he is again, that eye, wing, leg. All right, let's watch that again. That little heartbeat beating away right there. Again, this is inside the shell as this embryo is developing. Getting a little bigger right there. Even bigger, squirming around right there. All right. So day 3 is where we have the beak and the wings beginning to form. And again, here's our zygote right here. This whole area right here is all of the circulatory system that we see. And then here's our little embryo. And this little darker red spot right here is the heart. And so we're starting to see those, well, we can't see it, but the wing buds are starting to form and the beak is starting to form at day 3. Day 4, we're setting up camp. The tongue begins to form and the embryo actually turns on its left side. So if you noticed in the video, that embryo was laying on its left side as it developed. And so it will continue to lay on its left side for the duration. So again, here's our little embryo zygote right here. So this is the spinal column right here. And then there's that heart again. Day 5, the reproductive organs begin to form, differentiating male from female, and the bones of the legs begin to form. Day 6, the beak becomes visible and the ribs begin to appear. So again, here's that wing bud that we talked about earlier, starting to form. There's the eye, and the beak is starting to form on day 6. Day 7, the leg is able to bend at the knee. Again, here's our little, zygote right there, our embryo. And again, when we look at this whole entire circulatory system, it's taking over like half the yolk already at day 7. And again, that's all circulatory system feeding that embryo. Day 8, the feathers begin to form. So here's that little eyeball, wing bud, leg. Day 9, the mouth opening appears. So when that beak forms, it's formed as one section, and then at day 9 we start to see that separation of the beak, of the top and the bottom. So again, here's the eye, wing, leg, and that yolk. On day 10, the beak starts to harden, and we can start seeing the egg tooth. So right here, this little white speck at the end of the beak is the egg tooth. So here's our beak. Here's our little legs. All right. So what's that egg tooth used for? The egg tooth is this little tip right here at the end of their beak, which falls off shortly after they're born because it's not needed, is essential for the pipping or initial breaking of the shell. So when the egg, or when the chick starts to hatch out of the egg, it uses that egg tooth to actually break the inside of the shell, and that's called pipping. So day 11, we see the development of the toe claws, and they begin to curve downward, and then the bottom of their feet become padded. That fatty tissue that pads the bottom of their feet starts to appear. So we can see that right here. So here's that beak, big eye, arm, here's that leg, and here's those toe claws. So on day 11, we actually start to see those starting to curl down, and then the fatty part of the bottom of there starts to form. Day 12, the ribs begin to ossify. The word ossify means to become hard, so it's not as jelly-like in substance, I guess. And so they start to become hard like bones. Day 13, starting to look like a chick, right? So on day 13, the left and right collarbones fuse to form the wishbone. How many of you guys have ever, like at Thanksgiving, taken the wishbone and broken it apart to see who wins to make the wish? So on day 13, that wishbone is actually formed by those left and right collarbones fusing together. So again, here's that egg tooth that we talked about earlier, the big eye, and then we have the wing here, and then we're starting to see those feathers right there. So on day 14, the embryo turns its head toward the blunt end of the egg, where that air cell is, and the skull begins to ossify. Again, ossify means to become hard. Okay, so again, we still have that egg tooth there. We got some feathers, our toes, and our wing is tucked in there somewhere. On day 15, the small intestines are actually, so the small intestines are actually in the yolk, and the yolk is being used for the nutrition that the chick needs to develop. And so up until day 14, the small intestines are actually inside the yolk and not inside the abdomen of the chick. And so on day 15, those small intestines actually are taken into the body from the yolk. Day 16, all feathered up with no place to go yet. The chick is fully covered with feathers now. On day 17, the beak turns towards that air cell. And then days 18 and 19 are they kind of blur together, but as far as what's happening, but the beak actually breaks through that inner shell membrane, and the lungs start to practice breathing using the air cell. Day 19, the yolk sac actually enters the body cavity. So again, we talked about the intestines moving in on day 15. On day 19, the whole yolk, actually what's left of it, as we saw it getting smaller and smaller, actually enters the abdomen, and the abdomen sutures up or closes up with the yolk inside. And then the chick is going to use that nutrition from the yolk to hatch out of the egg and shortly after until it eats. So that yolk is the source of nutrition for the chick. Day 20, the lungs are using the air cell for breathing completely, and he decides, or she decides, that, hey, this egg is too small for me, and the neck starts to spasm, and then it uses that egg tooth to pip through the shell, and eventually, day 21, exhausted and wet but alive, the chick is hatched. So if there are any questions up to this point, go ahead and put those in the chat box, and we'll try to get those answered for you. But for the sake of time, we'll move on. So this next part that we're going to do is we're going to actually look at the egg, dissect open the eggs, and let me get that picture for you. Oh, picture, sorry. Everybody should have access to this form or this worksheet, and this is what we're going to go over when we do our activity. Okay, so we're going to actually dissect open an egg and look at the different parts and talk about those functions as we go forward.
All right, let's go ahead with our activity. Like I said earlier, you should have this worksheet or have access to this worksheet, and if not, that's all right. What we're going to do is we're going to go through this worksheet and identify the different parts that we discussed earlier on an actual egg. If you'll notice down at the bottom, you do have a word bank that you can use, and so when we fill in these blanks with those words, you can go ahead and cross them off. If you're too little or it's hard for you to get them written in the blanks in the time that we have, you can go ahead and just draw a line from the word to the location or to the box instead of writing out the whole word. So a modification for the younger kids. All right, I'm going to go ahead and switch this out and pull in our egg. So what you should have is a plate, and if your egg's not cracked yet, that's fine, but you'll have a plate with an egg. Go ahead and crack your egg open if you have not done that. So if you need a parent to help you with that, parents, go ahead and help, and try not to break that Vitalin membrane around the yolk yet. So I'll give you a second to go ahead and crack open your egg. All right, so let's go ahead and get started. I'm going to use a toothpick to help identify the parts, and I might have to move my camera too as we go along just to get closer up on the images. So that first thing that we notice is the yolk. Again, this entire yellow portion right here is the yolk. Again, it is high in vitamins, minerals, and fat. And again, the embryo is what is going to use the nutrition within that yolk while it's developing, and then again right before it hatches out. And the hen is actually forming this yolk during the day. Then we go down to the germinal disc. And I'm going to go ahead and take my phone off my stand here and see if we can get a close-up of the germinal disc. So the germinal disc is actually this white dot right here. And that's where fertilization is going to occur and where that embryo is going to develop. Okay? So try to identify where your germinal disc is. It's just a white speck on top of the yolk. Okay, so we see this little round speck right here. That's the germinal disc. So on your worksheet, you can go ahead and fill out that on your worksheet. All right, and then next is the Vitalin membrane. That's kind of a hard word, so try to say that out loud. Vitalin membrane. And that's the membrane that holds the yolk together. And so again, when you break the yolk, or that's what you say, you're actually breaking that Vitalin membrane. Okay? And then as we go down our worksheet, the next thing on there is going to be, it's pointing to the rope-like structure on the ends of the yolk. And again, that's the Kalasa. It's the Chalazae word down at the bottom on your word bank. So find that word and either draw a line or write it. And I'm going to move my camera once again. And the Kalasa, move my light out of the way, a little glare. The Kalasa is the white, cloudy portion at both ends of the egg, or of the yolk. So there's that side over there. And then on this side, it's right here, kind of underneath the yolk. There, it sticks out. And that's the Kalasa. That suspends the yolk inside the shell so that it doesn't rest on the sides. Okay. So again, that's the Kalasa, the rope-like structure. Moving down the worksheet, the next one is the shell. And so we can look at the shell here. This one is a brown egg. We raise chickens on our farm, and so we have brown eggs. We have hens that lay brown eggs. The color of the shell is dependent on the breed of the chicken. So store-bought eggs are typically white, and that's because of the breed of the chicken that lays those eggs. So eggs, the shells can be brown, green, blue, speckled, all kinds of colors. And again, the shell has hundreds and or thousands of tiny little pores that exchange gases, carbon dioxide out, oxygen and moisture in, that we can't see. And then if we go to the top of the worksheet, it is the air cell. So that's what's pointing there. And let's see if we can, I don't know if we're going to see this in the light, but if you pick up the end of the shell, there'll be a pocket of air. It's just a little bubble. You just kind of see it popping up there. That's the air cell. And so that's what the chick is going to use to breathe before it hatches. So it's just a little pillow of air inside that shell. Oh, there we go. There you can see that little pocket of air a little better. Okay. So again, that's the air cell. Moving down the sheet, next are the membranes, the inner and outer shell membranes. And this surrounds the albumin. It provides that protective barrier on for the egg. And that's what the air cell forms in between, are those two, the inner and the outer shell membrane. So in that box, you'll put the shell membranes. And then as we look at the albumins, or the white part of the egg, when it's fried, that's what turns white, that's what's clear here. We have two types of albumins. We have a thick albumin and a thin albumin. So nearest to the shell is the thin albumin, and as we can see on our plate, we have a much thinner liquid here, and then we have an actual frame where it looks thicker and more like a pillow top. Okay, we notice the difference in the thick albumin versus the thin albumin. All right. The thick albumin is this part in here inside this shell membrane, the inner shell membrane, and it's closest to the yolk. And it's high in riboflavin and proteins, and it's what that chick is going to use for nutrition for those two nutrients. The thinner that this lays out, the thick albumin, is the lower the grade of egg it is. And so we want a thick pillow top when we crack open eggs to see that they're high quality eggs. All right, are there any questions? If there are some questions, go ahead and put those in the chat box. And You can go back and view the video for the answers if you missed one. Again, the words are on the bottom of your worksheet. And we'll go ahead and look at the answer sheet for your worksheet. So these are the answers. Sorry, my light is gonna have a little reflection on it. And then I also forgot to mention that the albumin, we talked about the yolk being formed during the day. The albumin is actually formed during the night when the hen is forming her egg. So again, we have the yolk, germinal disc, vitellin membrane, Kalasa, shell, air cell, shell membranes. Thin albumin is closest to the shell, and the thick albumin is closer to the yolk. I hope you have learned something today about either the development of the embryo or the parts of the egg and their functions. If you have any questions, again, put it in the chat box. A homework assignment that you can do during time away from school is to get a bottle of vinegar, just straight vinegar, pour it in a cup, place an egg inside the vinegar, let it set overnight, and then in the morning or later in the day, the next day, go ahead and pick it up out of that vinegar, wash it off a little bit under some running water, and then refill your cup with some more vinegar, fresh vinegar, and then do it one more day and see what happens. If you do this, I would appreciate some pictures of the final product. You can email those pictures to me at smithheather@missouri.edu and show me what your final product looks like and give me a little description of what you found out about it. Again, tune in tomorrow for another lesson on 4-H online, and we will see everybody's pictures hopefully soon. Thanks.